Right. Thank you and welcome back to another HPL seminar. It's my pleasure today to introduce PhD candidate Tim Vanderzee. He's under the supervision of Dr. Art Quo. Tim is from the Netherlands, if you didn't know that already. He did his undergraduate and his master's degree at the Free University of Amsterdam, and he studied biomechanics. As we were just talking about, he's now been working on explaining the problems of muscle models for 10 years at this point, so it should be very fun today. So his thesis in his undergraduate, for his undergraduate degree was using muscle models to explain metabolic cost of steady state force production, and now he's moved into doing cyclic force production, but with the same premise. The current research interests obviously are understanding the mechanisms behind the mechanics and energetics of force production during human movement. And his proudest scientific accomplishment is that he received the Paper of the Month award from the Journal of Experimental Biology in October 2021 for the paper titled Soft Tissue Deformations Explain Most of the Mechanical Work Variations of Human Walking. His future plans are to implement more mechanistic muscle models and whole body musculoskeletal simulation to better predict the metabolic cost of movements as part of his postdoc position. I'll let him explain that more. Aside from this, outside of work, he's a competitive tennis player. He's been playing for the Dinos for the last 5 years, and he's also a tennis coach in his spare time. Tim's a brilliant scientist. If you've had a chance to talk with him, it's always super fun and interesting. So I'll shut up and turn it over to him now.
Thank you, Michael. All right, so as mentioned, I will be talking about the missing piece in models of force development in muscle. So typically in biomechanics, we are interested in movements that are cyclical, and we can measure things like excitation of muscle and biomechanical signals like the joint angles and the ground reaction force. And we could take a systems approach and relate excitation to biomechanics through muscle, where muscle takes excitation and produces force, acts on the body, and via Newton, you get things like kinematics and stuff like that. And important to note here is that both the body and muscle can be treated as dynamical systems, with the link between them being muscle forces, one of the links, of course. And the problem we typically have is that muscle forces are known during movement. They're not easy to measure. And this is a shame because muscle forces could be informative for many aspects of movement science. It could inform us about bone stresses. It could inform about motor control, the relationship between neural signals and how muscles produce force. And muscle force production also has a metabolic cost. And so if we would know the muscle forces, we would also be able to better predict metabolic cost. And so a typical approach we have in biomechanics is that we model this using musculoskeletal modeling. And typically what we do here is we take hill-type models as a replacement for muscles and connect these together with a skeletal model in a framework like OpenSim. And of course, the muscle forces are not the actual forces, but they're predictions. And so there's a question of how good these predictions are. And how good is this hill-type model? Well, the hill-type model has many issues, which some of them are already discussed in the literature, and I will just explain these now. So one thing is that the hill-type model has been used to predict metabolic cost of movement, and here we see a diagram or a graph with measured metabolic cost on the vertical axis and hill-type predicted cost on the horizontal axis. And then quite a lot of studies show that the hill-type model can actually predict the cost quite well. So for level walking, we have a predicted cost that's close to the measured cost. But the model is not that great in predicting what happens if we change conditions. So here is in green, what happens if you change the incline of walking and you walk uphill. A recent study by Koolewijn showed that then the hill-type model only predicts about 38% of the metabolic cost. Another example is the Hill-type model as it's used to estimate muscle excitation. Here's an example of the muscle excitation during the first half of a hopping cycle, a recent study by Jessup et al. And then they tested the Hill model prediction, and well, they say that it's quite good, but there are some differences. So one thing is that it looks good, but that's sort of fake, because in this graph the amplitude is made the same to facilitate comparison, and that's a very typical thing where we have a normalized excitation on the vertical axis and we don't actually test the amplitude, and it's sort of, well, you get suspicious and you might think that the amplitude is actually bad, and that's why we don't report it. And then another obvious thing is that the timing is wrong, and this is just one example, but you can find multiple examples where typically the Hill-type excitation is too late. All right, so then there's another type of experiment where we have standing and we perturb the surface and then observe the reflex response of muscle. And then you can typically observe an increase in the torque. This is hip torque in this example as a response to that perturbation. And it's typically described as short range stiffness. And then the Hill-type model has a prediction of force that's too low. And then recently in a review by Yu et al., it's also shown that the Hill-type has a negative stiffness on the descending limb of the force-length relationship, which means that it's numerically unstable. And there are some more fundamental problems with the Hill-type model, it being phenomenological, and it's using curves that are just fitted without a mechanism. And one of these is the force velocity curve. And the problem with that is that force velocity in muscle is actually not an instantaneous property, but a steady state property. But this is not taken into account, where in the Hill-type, there's just a direct relationship between force velocity and activation. And obviously, movements are typically not steady, but feature cyclical length changes and muscle activation. So the question is of whether this is appropriate to use this kind of relationship. Okay, so the force prediction of the Hill-type model might not be that good. And if that's the case, then of course that limits many aspects of movement science where we would benefit from accurate force predictions. And so in this talk, I will argue that there's a missing piece in the Hill-type model that may explain why these forces are not, the predictions are not that good. So here's an outline of the presentation. I'll start with discussing some of the muscle force development dynamics, including twitch summation and cyclic muscle contraction. And these are typically not included in many muscle models. Then I will talk about muscle models and their prediction. I will talk about the Hill-type model, and I will explain that its dynamics are insufficient or misattributed. And then a typical alternative to that is cross-bridge models. And surprisingly, I found that cross-bridge dynamics are also insufficient to account for force development dynamics. And then finally, at the end of the presentation, I will talk about a model that model of this missing piece. All right, so let's consider the muscle as a system where an action potential is the input and we treat force as the output. Here is a schematic of the muscle where you can see the muscle fiber, and we can here see an action potential that travels down the T-tubuli, and then when it hits these receptors, it triggers the release of calcium from the sarcoplasmic reticulum into the myoplasm, and then it can diffuse through the muscle fiber. And then, of course, it can interact with the myofilaments through its attachment to troponin. And here we see how, well, a simplification of how calcium binds to troponin. And this is, here I define this as calcium activation. And you can try to measure this as the relative amount of calcium bound to troponin. And important to note here is that that is fast. So it's thought to have a peak value in about 5 milliseconds after the action potential. And then once calcium is bound to troponin, the cross bridges can interact because now the binding site on actin is exposed, and a myosin can attach and produce force. But then, maybe quite surprisingly, this force is way slower than the calcium. And the peak force of a twitch is typically around 100 milliseconds after the action potential. And so there seem to be dynamics in between, but it's maybe not necessarily clear what those dynamics are. Also important to note is that this cross-bridge cycling is associated with an energy cost, as there's ATP required to detach the cross-bridge. And then another thing here to note is that the decay of calcium activation is quite slow. And the reason for that is that the pump is, well, quite a lot slower than the release. And so calcium eventually is pumped back during relaxation to the sarcoplasmic reticulum, and that also costs energy. All right, so here we have our diagram of muscle where I can see that we have a fast calcium activation, and then there's some slow dynamics before we get some force. So what happens if we do not one action potential, but multiple action potentials? Well, we can then define a firing rate. It's the 1 over the time between the action potentials. And you can see a response in calcium activation approximately looking like that, and a twitch summation looking like this. And then you can plot the peak force versus the firing rate. And you can also do that for the twitch and for a very high firing rate. And then you get a typical relation like this. This is actual data of human quadriceps, which is sometimes called the frequency force relation. And so one of the questions I had is, how can we understand this relationship? And I thought of second order dynamics because we have two distinct processes in force production that I described. We have calcium activation and some force development that's happening after. And so here I have this simple dynamics where we have some excitation and then activation that represents calcium, where we have two different dynamics for forward and backward, because forward is the fast release, backward is some slow pumping. And then we have some dynamics between activation and force. And so I show here some taus, these are time constants, and I'll quickly explain or give a refresher what that means. So here is an example of a step input. So that means that we just make the excitation equal to the maximum value for some time, and then equal to 0. And then you get a dynamical response in activation that's looking like that, and it decays like that. And a time constant, right, that's a step response. Time constant is just a quantification of how fast that is. So the time constant here in this case is the time it takes for activation to reach about 63% of its maximal value. And similarly for deactivation, it's the time it takes to go back to 37%. And in this case, we do second order. So you can see that as two first orders in series. So it's a little bit more complicated. So we have discussed this relationship, and then this relationship is similar, but now activation is the input for that relationship. But important to, just the summary is time constant is a way to quantify how fast dynamics are, where a larger time constant means that the dynamics are slower. And so here I just fitted these three time constants on this relationship, and I found that what you get is sort of what we expect, where the activation time constant is very small, then the deactivation is somewhat larger, and this force associated time constant is very large. Okay, so we can use those as just a ballpark estimate of our dynamics. Then this is great, but in movement, you typically have bursts of excitation, not single action potentials. And well, you can measure that with EMG. And then you can actually quantify here a different type of frequency, which is the frequency between bursts, which could correspond, for example, to contraction frequency. And then if we analyze EMG, what we typically do is do some rectification and smoothing, and then you can maybe get an estimate of some kind of overall muscle excitation. The amount of excitation you can quantify as the amplitude of this thing, AU. And you can also measure a force with a certain amplitude, AF. And so then you can quantify a gain of this system, what's the ratio of these two amplitudes. And the second order dynamics would indicate or suggest that this gain would decrease with cyclic frequency. There's another type of estimate or a measure we can take from this, and that's the delay between excitation and force. And again, this dynamics would predict that this delay increases, and this is a negative axis, so it goes down. So is this cyclic contraction also consistent with the second order dynamics? So just to see if we have some more proof for this idea of second order dynamics in muscle. Well, to test that, we use some old data that we collected in a cyclic force experiment here at the HPL, where we used the Baudex downstairs where participants were seated. We used isometric mode, so there was no movement. And then we asked participants to produce sinusoidal torques while matching their torque against with the target displayed on the monitor. And these torques had variable frequency, so this is variable cyclic frequency, but constant amplitude. And then we also measured the EMG. And then so we can then relate torque to EMG, and torque can be an estimate of force. And we can try to get these relationships. So here are some average data. On the top panel is torque versus time. Note that there's different scales here on these axes. But you can see that they're both sort of look the same, meaning that they have the same amplitude. And of course, we have a higher frequency there. And then on the bottom is EMG, where these do not really look the same. The EMG with the high frequency condition has a higher amplitude. And this means that there's a decrease in gain with cyclic frequency, and that's roughly matching this second order dynamics. And so this is, just to clarify, these dynamics, I'm using the same time constants that I fitted on the action potential twitch summation. So that's quite cool. Then for the phase delay, the predictions are a little bit further off, but it still roughly captures the data. So overall, it seems that muscle behaves like a second order dynamics system, which it indicated from both the cyclic contraction data and the twitch summation. And as I said, there's a little bit of difference here at higher frequencies in the phase, and that could be explained by a second order being, well, the actual muscle behaving like more than a second order, maybe third or fourth order. So I would say at least second order. All right, so the first part, we've discussed the twitch summation and cyclic muscle contraction, and explained that this is consistent with second order dynamics. And you can think of this as one order is due to fast calcium and the other due to slow force development. That's just to give an idea of the basics of muscle contraction here. And this is just a diagram to show that in a simplified manner. So we have time here on the horizontal axis, and the idea is little time required for calcium and a lot of time for force development. So now let's consider the muscle models. As I said, it will show that the Hill-type model dynamics are insufficient or misattributed, and that the cross-switch dynamics are also insufficient. Okay, so this is where we are right now. Second order dynamics, things work, that's good. But of course, muscle is more than just second order dynamics. We also have properties like the effect of cross-sectional area, the interaction with elastic tissues like tendon, the force-length relationship, and the force velocity relationship. So we would like to put these in our model. And I'm just going to call them steady properties here. And if you do that, then you almost created a Hill-type model, actually. So yeah, things are not that different. So what about these dynamics? So let's first consider the Hill-type force dynamics. I call these muscle tendon dynamics because they arise from the interaction of force velocity with the tendon force length curve. They're entirely dependent on these two relationships. For previous work, I estimated the force length curve of tendon from ultrasound. So this is for my setup with the isometric Baudex. This is ultrasound of vastus lateralis, where I have this TIMTRAC algorithm that estimates the fascicle length. And from the fascicle length, you can also estimate the shear elasticity, and you can we made this curve. And then the other property that's important for this dynamics is force velocity. It's quite tricky to do this. I took some literature value, previous reported data with electrical simulation, and got this force velocity relationship for the quadriceps muscles. Okay, so if you combine these two, we can now evaluate the dynamics associated. So this is an example of a step in activation. So we set activation to the maximal value, and then you can observe the force response. And this doesn't behave like a first order in force because it's actually length dynamics, but we can approximate it by just evaluating the time it takes for this force to reach that 63%, to just get an idea of roughly what how slow these dynamics are. And the answer is it's for this type of quadriceps curves, it is 55 milliseconds. It's about 55 milliseconds. Okay, so it's quite slow. It's not super slow. It's maybe sort of in between, medium. What about the activation dynamics of the Hill-type model? Well, to some degree, these are free parameters, so we can just choose these. Of course, there's values in the literature, but there's quite a lot of range in them. So the range is about 10 to 100 milliseconds for the activation time constant. So here we see the result for all of these time constants, where you can see that the prediction for the force firing rate, the twitch summation, is better for smaller time constants. And that makes sense because this twitch summation depends on fast calcium. And this is the calcium, sort of the excitation dynamics. So we want to get as close to that as possible. But then interestingly, for cyclic contraction, we have the opposite effect. So again, the dark ones are the fast, the slow, small time constant, and these are now way off, whereas the larger time constants are now indicating a good fit. And so, yeah, it seems impossible to predict both relationship with the same, with a consistent set of parameters. And also important to note here is that these large time constants, you might be tempted to choose them in order to measure cyclic contraction data, but they don't agree with calcium. That means that if you choose them, then the activation of your model doesn't agree with calcium anymore. And this is problematic if you want to estimate metabolic cost, because calcium is a contributor to metabolic cost, as it costs calcium or energy to transport calcium back during relaxation. So, okay, so the overall conclusion here is that we cannot simultaneously explain cyclic contraction and twitch summation with the Hill-type model, even though it is a second order. And you could say it is because this is slow, but not slow enough. And so we cannot fix it with our activation dynamics. And here's just what happens for default values, where typically 15 milliseconds, it's a pretty typical value. And it's sort of a compromise between the two, where they're not as bad as they can be, the predictions, but they're still quite far off. And this relates to what I discussed earlier, is probably in this simulation, this 15 milliseconds was used. And so this relates mostly, I guess, to the cyclic contraction, where you can see here we have an underestimation of the phase delay in our model, and that agrees with this timing error. And I would suspect that you could also see this gain error, but of course, it's for this data, it's not possible because they normalize the amplitude. So yeah, default values of the Hill-type model make it too fast. The gain decrease is too small and the phase decrease is too small. But that might make sense to some people because Hill-type models do not include cross bridges, and cross bridges have dynamics associated with them. So you could think that maybe they're too fast because they miss cross bridges. And the fact that they don't have cross bridges relates to another problem that I discussed in the introduction, is that their force velocity curve is just a curve fit, whereas muscle actually has dynamics associated with force velocity, which are the cross bridge dynamics. And this might also relate to some other aspects, as I discussed that cross-bridge dynamics are associated with an energy cost. So the fact that there's no cross-bridge dynamics might explain that the metabolic cost predictions is off. And recently, it has also been proposed that the fast increase in force, that short-range stiffness, could also be explained by cross-bridges. So many of the problems of the Hill-type model may be because it doesn't include cross-bridges. So yeah, that's why I thought to discuss them. So what we can do here is just add the cross-bridge dynamics to the Hill-type model and then leave those steady properties and the activation intact. And then that's very similar to just a cross-bridge model, as for example, Kuhn-Lemaire described in his thesis. But how does that work exactly? And how fast are these cross-bridge dynamics? Well, so one thing is that, as you might have missed, the force velocity jumped out of this steady properties because it's not a steady property anymore. It's a consequence of the cross-bridge dynamics. And then this is also a consequence of cross bridges, although we use the same tendon force length and we can make the cross bridges to have the same force velocity curve. And so this sort of effective time constant might still approximately stay the same. Okay, let's discuss about the cross-bridge model. This is from Huxley. This is a typical way to model cross bridges, where you have this idea of a cross bridge on the myosin filament that can attach to some actin binding spot within a certain binding region. And then the cross bridge has a certain strain, as shown here as a spring. And then what we are doing is we're using the distribution moment model, which is just a version of this where the distribution of attached cross bridges is approximated with a Gaussian. So just, yeah, maybe explaining this. So here you can see the strain. So there's a larger strain towards the right, and then the vertical axis is the relative amount of cross bridges that are bound for that strain. And this is the case for isometric contraction. So we can see that there's sort of this moderate strain in the cross bridges. And just an important note is that this distribution moment model only requires 3 extra states to quantify this Gaussian, the force, the stiffness, and energy. And this means that it's its computational cost is only a little bit more than for the Hill-type model. And this contrasts with some other cross-bridge models which can have a very high computational cost. Okay, so during shortening, the distribution shifts to the left to lower strains. And it also shifts down, meaning that fewer cross bridges are attached. And this is because we have these Huxley rate constants where the detachment rate is larger at lower strain. And for lengthening, we have a shift to the right, so an increase in the strain, but also a decrease in the relative amount of attached cross bridges, again, because we have a relatively high detachment rate. And then the attachment is relatively high in the intermediate region, the binding region. Okay, it turns out that this rate constants, these functions map to force velocity. So here in this animation, you can see the effect of the detachment rate at low strains. And there you see the force velocity relationship. And as you might see, what we're doing is we're shifting this intersection with the horizontal axis towards the right and left. Another way to say that is that this detachment rate determines the maximal contraction velocity of muscle. Another thing you can do is change all the other rate constants. And then what happens is something different is this maximum contraction velocity stays about the same at 0.6, but the shape of the force velocity relationship changes, which is called the curvature often. And there is sort of a one-on-one mapping of all the rates on this force velocity relationship. You can visualize that in a rate constant space where the attachment rate is on the horizontal axis and the detachment rate on the vertical axis. And then there's one point in this space that matches our force velocity relationship. And turns out for our empirical data here, we need 300 per second, 300 hertz for attachment and 1500 for detachment, approximately. But then another thing is that the overall dynamics of cross-bridge force development, which I call the tau c, is about equal to 1/f. And so in this case, that's about 3 milliseconds, which is quite small. Okay, so this means that we cannot freely choose our cross-bridge dynamics, the speed of our cross-bridge dynamics, because we have to fit on force velocity. So what about the activation dynamics? Well, the distribution moment model, typically these are chosen to match calcium, which I think is a good idea if you care about metabolic cost, like I do. And so these values, you might remember, are 1 millisecond forward and 50 backward are in agreement sort of with calcium. If you do that, then you get quite a good twitch formation graph, but you get a very bad cyclic contraction prediction. And that might make sense because we have now made these agree with calcium so that these are very fast. These are very fast because they have to agree with force velocity, and these are not slow enough. So just let's imagine that we choose different rate constants for our cross-bridge attachment. Well, then you could choose them so to sort of match this cyclic contraction. And then you would also match the twitch summation because we have good calcium parameters. But then, of course, the problem is you don't match force velocity anymore, as this is, I don't know, about factor 3 or 4 underestimation of maximum contraction velocity. So it's either or, and you cannot simultaneously fit force velocity and force development with cross-bridge models. And this is something that's super new, so I'm quite excited about this. Okay, so the summary for this second part is we talked about Hill-type models, that they are not sufficient to explain the overall force development, or they do some misattribution. And cross-bridge models, because they have to be fit on force velocity, are also not sufficient. So this is the summary of that. Hill-type models typically try to make activation a little bit slower. And so, yeah, you can see that here, but their force development dynamics are quite fast. Cross-bridge models have more force development dynamics because they have cross-bridge cycling, but both are too fast to agree with data. Okay, so what is missing? So we don't know, but we have a solution, and that is to put dynamics in between calcium and cross-bridge cycling. And for now, I call these force facilitation dynamics. And force facilitation, together with muscle tendon cross-bridge cycling, make up force development dynamics, and therefore that is sort of consistent with data. But what is force facilitation? Well, what do we know? We know that calcium activation ends when calcium binds, and that's very fast, 1 millisecond. And we know that the force production ends sort of when the cross-bridge attaches and produces force. And here are ballpark numbers that, yeah, I've been talking about during this presentation. Okay, cross-bridge cycling starts when we are ready to cycle, but the cross-bridge is still detached. But what happens in between calcium being bound and cross-bridges being ready? Well, this is not that clear. So this is sort of speculation what I'm doing right now. But one thing you can imagine is that there is this state where actin is activated, meaning the tropomyosin moved, but myosin is still not ready to produce force. And then between this and calcium being bound, the tropomyosin has to move. So tropomyosin movement might take time, might have dynamics associated with that, and there's X-ray diffraction methods to estimate tropomyosin movement. So here's an example. The blue dots are estimates of moved tropomyosin, and the red dots are force over time. And as you can see, the tropomyosin estimate isn't instantaneous. In this case, it takes about 40 milliseconds. And then in between, after tropomyosin moved, you could think maybe myosin still has to do something. And here's some data showing, again, X-ray diffraction estimate of myosin movement in blue. And again, this is not instantaneous. This takes some time, about 60 milliseconds. And of course, it's a little bit difficult to relate this to humans because this is non-human muscle and different temperatures, different conditions. But we get the overall idea that these things might need time. And what are the mechanisms behind this? Well, it's actually I think quite recent that people think about this myosin movement. And here from this study, there is a model fits to this data, where the idea is that, so this is the traditional idea is here, where myosin can be attached in red or detached in yellow. And then this new idea is that myosin can also be folded back, and that's sometimes called super relaxed. And in order to produce force from rest, it would need to move away from that back folded position. And these are model simulations where this green one corresponds to the relative amount of myosin that's in that super relaxed state. You can see it takes quite some time here to get out of that. Okay, so these are just speculations on what might be including in that, but for now we're approximating them with simple dynamics to match our data. And then one prediction of doing that is that these account for a big chunk of the force development dynamics, meaning that cross-bridge cycling and muscle tendon dynamics are relatively fast. And there's actually some ways to test this in humans where you can compare the rate of force redevelopment after a quick shortening to the rate of force development from rest, which you could do in a setup like our setup, where the prediction would be that force redevelopment is much faster than force development, because force redevelopment would only rely on cross-bridge dynamics, and force development would rely on the whole thing. Okay, so for now, what we did is we had our cross-bridge model. We added these facilitation dynamics just as a simple first order with that time constant. And then my current name for this is Integrated Dynamics Muscle Model. This integrated model fits force velocity fine because cross-bridge dynamics are left intact. And because you can have slow dynamics of these facilitation, you can also fit cyclic contraction quite well. And then because we have these fast calcium dynamics in combination with the other dynamics, we can also fit twitch summation quite well. Okay, so next step would be to see if we can validate this model more, but also include it in movement simulation and incorporate it in a whole body framework and predict cyclic movement. And so I haven't done that yet, but here's a first attempt. So this is a simple pendulum. So I connected this muscle model to a pendulum, let it drop and let it produce force, and you could quickly see that just to get started. And then I just wanted to point out that the computational cost was actually quite small. As I noted earlier, it's only a little bit more than the Hill-type model cost. And so that gives an idea that it's actually feasible to do these types of modeling in whole body simulation. All right, so that's what I have. I will just end with some take-home messages to summarize. First is that, as we observed in the first part, muscle contraction features fast calcium and slow force development consistent with second-order dynamics shown here. Hill-type models are either too fast or they misattribute slow dynamics to calcium activation. And so in this diagram, they're too fast. And so you can still use them, but if you use them, be aware of their shortcomings and use at your own risk. Cross-bridge models have more dynamics, and so we initially thought that would solve it, but it turns out that the cross-bridge dynamics are actually quite fast if you fit on force velocity. And so this also doesn't explain force development as we measured. And again, so if you use them, you can do that, but at your own risk. And for now, we propose an integrated muscle dynamics model with slow force facilitation as the missing piece that accounts for some of these missing dynamics. And with that, I'd like to acknowledge my colleagues for feedback on this presentation and, of course, the funding. And I'm happy to take any questions.
Let's start us off with a question. Yes.
Nice talk.
Thank you.
So slow force facilitation, would you call it the myosin activation? Like you thought initially of actin activation. So is it that idea of myosin activation? So there's some force on myosin, the cross bridges out in this relaxed state, but this force would allow them to rotate into a position that they would attach, basically.
Yeah, good question. Good question. I'll just go back to that slide. So here I suggest there might be two things: tropomyosin dynamics, which you could call actin dynamics, and myosin dynamics. And I don't know the contributions of each of these. And I think it's, if you have ideas how to test that, I think it's very difficult to do in humans. So, but even in animals, it might be difficult. So it's a question of how much of these dynamics are myosin and how much are actin currently, I think. And then I think what you mentioned is that myosin dynamics are related to force, and this is true. I didn't mention this, but actin, as we know, is activated by calcium, but myosin doesn't have calcium binding sites. But the idea is that myosin gets activated by force. And so if you would do a next version of this model, you would have a feedback loop of force going back to myosin. And the idea of this group is that there's a small amount of cross bridges that are always awake, that they're never super relaxed. And these are the force sensing ones, and they wake the others. And so then you get these dynamics. Yeah, but for now we simplified and made them all the same. So you could say we don't include force-dependent dynamics, so we sort of attribute most currently to tropomyosin. Yeah.
So how do they wake up?
That's a good question. Yeah, I'm not too sure about that. Yeah. You have to think about that.
So for a lot of your comparisons, you primarily focused on the concentric portion of the force velocity relationship.
Yeah.
What do you expect would happen if you looked at lengthening? It's like things get pretty sketchy. But
So, okay. I did, so I did talk about, okay, I didn't, I guess, talk about it. But this is just an example what happens when you change rate constants. And I didn't show each individual example, but I did choose this rightmost detachment rate to match my eccentric data. So in that regard, I did do.
so like in terms of your, like how you say it's an improvement on models that don't account for the second order activation dynamics of the other components that you added. Like in comparison to experimental data, say where you're doing the cycling on the side X, would you expect, like presumably they were activating during short period of cycles. So would you expect that if you activated, the muscles were activated during lengthening and they were like break-like or like negative work per cycle contraction cycles, would you expect that your results would be different? Like, do you think that the benefit that you've added would be there if you had like lengthening contractions in addition to your
So what we did is fixed end contractions. So you have shortening and lengthening and you have zero work. So what would you think of like isokinetic setup where you do lengthening of the joint, like extension of the joint or something, or?
I'm not sure exactly for like how you do that setup, but like there's a lot of conditions in gait where muscles are active during lengthening.
Yeah.
And for like all the metabolic cost models for muscle, they're really like the area where they're the sketchiest, and Hill type models in general is during those lengthening contractions.
Yeah.
So just wondering how like your addition kind of fits in there, and if it also like if it helps with to the same extent with those lengthening contractions as with shortening.
Okay, yeah, so I wasn't so clear on what aspect you meant, but now you mentioned metabolic cost. I think there's some interesting things there. So I think in general, the Hill type model is quite bad with metabolic cost, and the both for shortening and lengthening conditions. And the fact that the problems with shortening, you can see that in the uphill experiment where you're doing a lot of shortening work, a lot of positive work, the cost is not predicted. And so in the cross-bridge model, I haven't shown this, but I did do simulations. It's quite good in predicting the cost associated with shortening work. For lengthening, it's a little bit different, and that is because both shortening and lengthening are associated with a high detachment rate. And so that's the traditional Huxley problem where if you just do that, then you overestimate the costs of lengthening. But then people have sort of, well, there's this idea that some of the detachment of cross bridges during lengthening is free. It's like a mechanical breakage that doesn't cost ATP. And so a lot of people fix that in cross bridge models by saying that those the detachment of cross bridges for lengthening is like 5 times cheaper, which is cheating. But if you do that, of course, then it works. But you need another parameter to do that. Yeah, and I'm not sure what I believe or what people believe, whether this mechanical breakage is a real thing. And of course, yeah, lengthening is, there's much unknown about it and how Titan is involved, of course. And like if Titan would play a bigger role, then that could also explain the lower cost of lengthening.
Oh, well, I got a question from Walter. So go ahead, Walter.
Can you hear me?
Yes.
Okay. Yeah, I was just wanted to get back to the suggestion that Venus made about you know, how you might increase the rate of force development and maybe in cyclic contractions. And I wonder, you know, one of the things that happen when you contract the muscle, there is a phosphorylation of the myosin light chains that then gives you more force per given submaximal calcium activation. Also, the force rise is much quicker than in an initial contraction. And I wondered if you had considered that at all, that you might have, you know, in cyclic movement, phosphorylation of the myosin light chains that then changes the attachment kinetics of the cross bridges that would help you getting a force quicker in a cyclic movement, a second, third cyclic movement than in the first one.
Yeah, so I'm not too sure about the mechanisms behind that, but might it be that those dynamics associated with that could be sort of included in our facilitation? Because our facilitation model would actually predict that the second contraction would have a higher rate of force development, because if the facilitation relaxation is quite slow, you would still be in a sort of relaxed state, sort of an activated state, and you would increase force.
Yeah, it would be very similar, because people argue that in a super relaxed state, as you said, they're folded back and tethered, whereas in the open or on state, they are relatively close to the actin, and that's what phosphorylation does as well. It brings the cross bridges away from the myosin backbone and brings them into the neighborhood of the actin, and then the idea has always been that then cross bridges can bind much faster, and phosphorylation of the myosin light chains occurs relatively quickly, and dephosphorylation occurs relatively slowly. So you would have something almost like calcium that comes fast and then decays slower. Phosphorylation occurs fast and then decays slower. But maybe I can ask.
So
yeah, go ahead.
So just in terms of our model, I said that there might be various things to contribute to facilitation dynamics. From what you say, it seems that phosphorylation is fast, at least for activating. So that would contribute, but not a lot. But then, of course, our facilitation dynamics here also have a relaxation rate. And so, as you say, phosphorylation might be quite slow. That might actually contribute to the relaxation of our conceptual idea of facilitation.
The other thing that I wanted to just hear your opinion on is, because you did a Cybex experiment, activate, deactivate, activate, deactivate. And then you compared it also to running, where you said, well, we want to look at walking or running. And what I always think about force development in running, that has to occur very, very quickly when you hit the ground, because there's a lot of weight to carry. And of course, we cheat there, because we activate our muscle 60 or 80 or 100 milliseconds beforehand. So I always thought of cyclic movements like running or walking. You pre-activate the muscle, your calcium activation of troponin C has already occurred, your tropomyosin has already moved, you already have weak binding of cross bridges, which doesn't produce force, but it produces stiffness. So then you hit the ground and there's a tiny bit of a stretch of a muscle, and you have this spring that's infinitely stiffer than it was 100 milliseconds ago, and that allows you to build up force really, really quickly, not because you change the activation dynamics, but you activate it way before you actually need the force. And we do that, in every cyclic movement that I'm aware of, where we need force really quickly at one instant in time. We pre-activate the muscles a certain amount to essentially, kind of what I would call stiffen the muscle. And now all of a sudden you have this stiff spring, you lengthen it a little bit upon impact, and force increases incredibly rapidly, which you would never, ever be able to do from a relaxed state.
Yeah, that's a good point. So as I sort of mentioned in this talk, force development is super slow, and yet humans are able to survive. And so we have found clever strategies to deal with our slow dynamics. And I guess that's one of the strategies where you pre-activate, so that you don't have to worry about the first half of this. So like calcium and facilitation, those are all set, and the only thing that's left is muscle tendon cross-bridge cycling, which can be quite fast. And then another thing is that I didn't spend too much time on, is that this 60 milliseconds is for my setup, where I had quite a lot of length change, about 2 centimeters or something. Because I was at a short length, a small angle, and Brian talked about this a month ago, of course, where you have a lot of length change happening because of slack tissue, perhaps. And then you have much slower dynamics. And so if you are somehow able to stretch your tendon to make it stiffer, then these force developments dynamics can be way faster also.
That's all I have for the moment.
Thanks, Walter.
Do you think that, do you know if there's any data on whether rate of force development scales with like animal or muscle size? Like if there's, like, so you accounted for delays due to like excitation to activation. But there's additional delays due to like from the start of activation to deformation of the tissue, which potentially would contribute to like rate of force development or delays in force. And then that potentially could be influenced by like size of muscle. So do you think that that would like your muscle or your model's performance would differ depending on the size of the muscle?
Yeah, good point. So this is not included in the model, but so tissue deformation is, I think, a big one because, you know, if you're a bigger animal, you might have more like fat tissue or whatever. So you might, if you produce force, that might give in, and that might contribute to dynamics. In terms of our setup, actually, the Biodex is not purely rigid, so that's also one of the reasons that we have slower force development. And then smaller animals might have less of that, and also smaller muscle, I guess. So typically in models, the mass of the muscle is neglected. I don't know if that's still appropriate for very large muscles, and I don't know, there might be more friction and whatnot, right? So yeah, but I'm not aware of specifics. Yeah.
Yeah, I'm just wondering, you know, people build prosthetic limbs, for example, that let's say you're an amputee and you're missing above your arm above your elbow, let's say. People will attach a prosthetic limb, maybe including an artificial elbow. You can control a prosthetic like this with, let's say, an EMG signal. But the problem is the correlation between EMG and force is sort of super fuzzy, and maybe it works in some very narrow conditions, but in particular in dynamic conditions, like you're talking about, often that relationship kind of falls apart or is sort of unclear. So people like Dan Ferris will just say, well, you know, the brain will figure out what you need to do to, you know, take that signal and do something useful with it. But I guess I'm just wondering if you had a model like this that could, you know, say use that EMG signal as a proxy for excitation, and be able to predict accurate forces from that, maybe you know exactly what the person is trying to do with that elbow.
Yeah. Yeah, good point. So I think Art sometimes mentions this, like decades ago, there was a lot of literature on the EMG force relation, and it seems that people gave up a little bit where too many studies showed that there's not a great relation. But yeah, I think that maybe we can try to revise that. And if we have a good model, including these added dynamics, we can try to see if that's actually true. And perhaps we can better predict EMG from force and vice versa. And then, of course, if you have that, you would need to simplify that relationship a bit. But then you could, yeah, implement that and use that at the start. And then it's not perfect, and the brain still needs to adapt a certain way. But maybe then that adaptation can be quicker.
Have you, or do you plan to test your model with more complex input signals to predict force, for example?
Can you give examples?
You've shown some examples with like single twitches or a train of twitches or something like that. Just imagine richer signals that you might see, and you know, how would the predictions of your model scale to those more complex inputs?
Well, what I did do is the cyclic contraction, where I gave a sinusoidal input, like in my experiment. So that's a good sign that both of those work.
Although that's isometric, right?
Yes, but as I said, there's a lot of length change. So yeah, that's a question. But yeah, for sure, it's a good idea to test it on as many different conditions as possible and be creative and think of variations. So yeah.
So I was just wondering, for your initial second order model with that cyclic motion, part of the reason it doesn't match well is you only took EMG from one muscle. Because it's not like it was just vastus lateralis that was active. It was the whole quadriceps group generating that knee extensor torque. So if you're just taking EMG from one muscle, you don't know what the other, you're just assuming the other muscles in there are doing the same thing.
Yeah, that's certainly a good point. I believe I did do more muscles, but I just for now just analyzed vastus lateralis, so I can always look at that. But you mentioned that the second order model didn't work, but actually
Well,
that first it's not a model, but like that first system worked quite well. So which one do you mean?
well, you said it wasn't perfect. Like you said it wasn't perfect there.
Oh,
towards
the
end,
phase, yes.
The phase, and like, could part of your phase issue be the fact that you're only evaluating one of 4 muscles that are actually doing it?
I see.
And that could be why that you need to somehow account for it. Like if you repeated this in, like, say, the triceps.
Yeah. Well,
Double
so
extension.
I think where do we have that plot? No, sorry, way back. I think if you do this for other heads of the quadriceps, the timing will look similar, and that's just from what I saw in data analysis. I should maybe show that and make sure that that's actually true. But of course, the amplitude might differ, as different heads might contribute differently.
Yeah, and
But the timing might be sort of similar.
It's also the rate of force development in each head is different. So,
Yeah.
from animal work we've done, rate of force development in the quadriceps group is very different for each muscle. So they don't all have the same exact rates. Some take a lot longer than others. Like in the rabbit model, vastus lateralis is really fast. Its rate of force development is high. And then it's rectus femoris, and then it's vastus medialis is the slowest for rate of force development.
Yeah. Yeah. So as I said, I think that in my case, all heads are roughly doing the same. But yeah, I'll make sure in a paper to include all the muscles.
I just wanted to comment on a couple things. So first of all, this graph right here, I think you might use to answer Ryan's question. So Ryan's question was, is there a EMG to force relationship? And I think when you show these second order dynamics and you show data, it does suggest that with some added force facilitation dynamics that you can do a better job with EMG to force. Is that correct?
That's true. But then the thing is, this is just one condition, and then this is like isometric at the joint level, and so yeah, for that case, yes, and for other cases, we still have to investigate that.
Okay, so just to give a plug to somebody else, Jeremy Wong has experiments on reaching, which is movement, not isometric, and he also gets a better EMG to force relationship by including something like these extra dynamics. Is that fair? It's fair. Okay, so that was just a comment, but also I want to comment on this question about animals of different sizes. And so first of all, most of what we know about muscle, like the vast majority, comes from like frog, rat, things like that, mouse. And so we don't have a lot of information about a lot of big animals, especially human. And so in our world of musculoskeletal modeling, a lot of the numbers that are used in those hill type models are basically fiction. And so even activation, people say activation dynamics as if activation is something you can measure. And usually activation is just used to talk about a fictional quantity that would be a nice intermediate that could help with force. And so we don't actually know. Sometimes people say activation means calcium troponin, which is sometimes a measurable quantity, but not usually in human. And then, so I just want to point out that what you're using is, when you're saying activation, you actually do mean something that is theoretically measurable in some cases, usually not human.
Yeah.
Okay, and then what about big animals? So how could we possibly know? Because even for human, a lot of this stuff is fiction. So basically, I don't know the answer to your question, but let me just speculate. So in big animals, almost everything goes slower. Nerve conduction takes longer because you're going a longer distance. You have bigger muscles, and so from the motor point, the spread of the electrical activity takes longer. So basically, everything you expect to be slower. So I'm just going to speculate. I would be shocked if there was anything about bigger muscles that sort of reverses that effect. Bigger animals have more time to do everything, and it's in my opinion, it seems like they use that time, and so I would just speculate that bigger muscles would be same or slower than smaller muscles. Maybe you can comment.
Yeah, I believe in general, bigger animals have also more slow twitch fibers. And so that would also contribute to them being slower. So yeah, I think the overall idea is that they're slower, so they would be suffering more from these dynamics.
So on the same line with diet, so you're talking EMG force, what about EMG calcium?
EMG to calcium.
Calcium release and all that, so there is a strong relationship between calcium release.
That's a good question. So that's not typically, or maybe never measured, because calcium is typically measured when there's single action potentials or tetanus, and EMG is measured in living animals, humans. But what I showed here in this part is that for both types of input, for both the action potential and EMG, I can predict the outcome force. And so it seems that, that suggests that the EMG should be compatible with calcium, just like action potential is compatible with calcium. And then in terms of how to predict that, so well, calcium is a state in our model, so we can treat our excitation as similar to EMG, or we can even treat EMG as our input. And then what you will predict is that, its timing is very similar because it's 1 millisecond time constant, and then its decay will be a lot slower.
Okay, go ahead, Walter.
Yeah, good. Thanks again. I just would like to follow up on a comment that Art made regarding large animals and small animals. We also know that in small animals we have, like in mice, you have a fast fiber type that doesn't really exist in big animals like humans, that are much faster and much more powerful in that sense than what we have in big animals. So that kind of further confirms that we actually have structural adaptations on the mice in heavy chain level that makes muscles in small animals quite a bit faster and quite a bit more powerful than what we have available. So that's a good point, like considering when you go from a mouse to a human. And then the other question that I had is a very, very specific one, a very small question. That is, you're using the force velocity relationship of Westing et al. 1990, the electrically stimulated one. But Westing also showed very nicely that the voluntary contraction, especially of the eccentric part of that force velocity relationship, looks completely different than the electrically stimulated one. And since you're doing, I think in the Cybex, you're doing voluntary contraction, is that correct?
Yes.
I wonder how you reconcile that. Why didn't you take the voluntary one that has a very distinctly different eccentric part compared to the electrically stimulated one?
Yeah, because that's a good question. The reason is that what's thought that okay, to just be clear, involuntary contractions, the eccentric force is less than in stimulated conditions. And I believe that's a self-saving mechanism for if humans would maximally activate, they would hurt themselves too often. So they have some inhibition going on where they are not allowing themselves to maximally activate during eccentric contraction. And you can measure that with EMG, and that's been done. And that's also true for isometric compared to fast shortening velocities. So in lots of regions of the force velocity curve, the idea is that humans cannot maximally activate. And then I'm just interested in muscle properties. So I just want to know, what if you would maximally activate? How would the force look? Because then I can, in my model, do a step response where I put maximum activation in my model. And so you could also use voluntary curves, but then you would have to do a submaximal input into your model to compare that to. And then that becomes tricky, like what value you're going to use. So then what you could do, for example, is try to estimate the percentage of maximal contraction that people did in those eccentric contractions and input that into your model. But you know, EMG might not be perfect one-on-one to excitation. So yeah, that's why I chose electrical simulation data.
Yeah. Well, talking about EMG to force predictions, you know, many years ago there, I think I mentioned that before, 25 years ago, we published a couple of papers on using EMG to force predictions in a cat hind limb that we trained with artificial neural network. And we actually got incredibly surprisingly good results in them predicting to other animals and other muscles what the force would be based on that trained artificial neural network. So I'm not sure what's there, but there's clearly something there that a pattern recognition algorithm can recognize and relate force very accurately to an EMG. So I'm sure there's something there that could be used if you're clever enough to figure it out.
Yeah, that's good to know. So that supports yeah, what I presented here. There is something that explains relation EMG to force. And so in contrast to neural networks, we want to try to express that with as little parameters as possible. So I tried to do that with second order, for example, because then like you can try to understand it. And with the neural nets, of course, you might be able to predict it perfectly, but you might be overfitted somewhat, and it might be difficult to extrapolate that to different animals, subjects, yeah, humans, etc.
Yeah, and that's exactly why we never really pursued that. Any earnest, because as you say, you know, that then spits you out the right result, and you don't really know what happens in those hundreds of nodes that are connected. You know, so that's exactly why I wasn't particularly interested in pursuing that. But I think there's something there that I think can potentially be very useful, and I think that would be good to pursue for people who have the interest in that.
Yeah, I'm very excited to try to do that in more conditions. And what Ryan said, it can be applied to all kinds of things, such as the prostheses that are driven by EMG. So yeah, it's an interesting topic, I think.
Thank you. That's all. Thank you.
Thanks, Walter.
Do we have any final questions? Okay, not. Well, thanks, Tim, again.